deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. DNA is located in the nucleus of our cells and our chromosomes and is made up of various smaller molecules called nucleic acids. It has a double helix that looks like a spiral ladder. Sugar and phosphate form the backbone of the ladder while the nitrogenous bases form the rungs. The sequence of these nucleic acids tells the cells in our bodies how to build specific features of ourselves like our nose, eyes, hands, feet, and everything else. If the sequence of something isn't there, our bodies would not have the ability to build it. The information in DNA is stored as the code name of 4 different bases: adenine, thymine, guanine, and cytosine. Adenine always pairs up with thymine and guanine always pairs up with cytosine. The human DNA consists of about 3 billion bases and more than 99% of those bases are the same in all people. As for personalities, imagine 2 kids. One is outgoing and social while one is timid and more self-reserved. Both exhibit completely different personalities. Why is this? The answer to this question is that our personalities have a genetic component. This doesn't mean that we are born with our personalities and nothing else involved. The genetic framework for our personalities can be manipulated due to our life experiences. Genes are not solely responsible for our personalities. Personality is a complex trait that is influenced by many different genetic and environmental factors. This is what we call nature versus nurture. Now let's move on to phenotypic traits, also known as our physical traits. Inherited traits as passed from parent to child through genes and chromosomes, which are made up of DNA. Some genes are dominant while others are recessive. Imagine if your mom and dad have dimples and you, their offspring, do not. You will need to have a serious conversation with them. With advances in molecular genetics, it became possible to launch the Human Genome Project, a sequencing project that determines the genetic makeup of an organism by reading off the sequence of the 3 billion DNA bases, AGCT, which encode all the information necessary for the life of an organism. The work of the Human Genome Project has allowed researchers to begin to understand the blueprint for building a person. It can help us understand diseases, including genotyping of specific viruses, treatment for diseases, identification of mutations linked to different forms of cancer, and the design of medication and more accurate prediction of their effects as researchers learn more about the functions of genes and proteins. This knowledge will have a great impact in the fields of medical biotechnology and life sciences with all these new advances in genetics that scientists are discovering. Have you ever wondered about the different changes we can make to ourselves if we could alter our DNA, such as if we could control the side effects of menstruation? Let's just say that we could be close to having that as a reality. Since scientists have now found a way to change the sequences of messenger RNA, basically rewriting the genome's instruction en route, making proteins, women with PMDD who experience major symptoms of menstruation now have a great chance to experience menstruating without the risk of dependency or addiction associated with conventional painkillers, all thanks to RNA editing. Rosenthal, a scientist, is currently working on editing the mRNA for a gene encoding the sodium channel NaV1.7, which controls how pain signals are transmitted to the brain. Permanently changing the NaV1.7 gene through DNA editing could eliminate the ability to feel pain and disrupt other necessary functions of the protein in the nervous system. Eliminating the ability to feel pain in the nervous system would allow women to experience pain-free periods, as the nervous system plays a very important role in temperature and pain throughout the body. In conclusion, I agree with this type of procedure because it could fight tumors that hijack important cell signaling pathways, those involved in cell death or rapid growth. If RNA editors could turn off signaling molecules even temporarily, the tumor could die and the patient could stop therapy, allowing the pathway to resume its normal function.